So what I'll do is I'll go in and take questions now. Anybody else have any queries? Anjali, you have put your hand up. Please go ahead.
Sir, what is endosome in the first slide that you showed?
So endosomes are essentially vesicles that are pinched off. That could come from the plasma membrane. There could be, they are brought in through endocytosis. And these now will carry, not just what's on the membrane, they will carry receptors. And the endosomes can get targeted to very specific locations inside the cell. The endosomes also have receptors which are bound to ligands. And so as they are brought in, they can be used for many different purposes. They can be used to affect the architecture of the lipid membrane as they keep, going in. They could also be, used to regulate signaling. They could be used to regulate the availability of receptors on the plasma membrane. All things that, that the endosomes are able to do. We didn't talk extensively about the process of endocytosis in this. There are different mechanisms of endocytosis as well. And, when you come to the advanced cell biology course, we will be talking about, clathrin mediated endocytosis, caveolar endocytosis, and such. Kishan, you have your hand up.
Sir, you were talking about how the stiffness of a cell can affect the gene regulation.
Mm-hmm.
So, if the cell shape was able to was manipulated from the outside,
Mm-hmm.
would that mean that we could change how the genes regulate?
Yeah. So, if you take cells like the endothelial cells that line our blood vessel, okay, and people have done these experiments where they put them on dishes, right, on slides, and all you do now is you grow them normally, and then they form a nice sheet of cells, right, and then you put them in a chamber where you can flow liquid at the rate at which it flows in your blood vessels. Okay, just having that flow of liquid, okay, above the cells, right? So the liquid is flowing over the cells, changes the orientation of the cells. So the cells are, kind of pointing in all directions. The moment flow starts in this direction, in a couple of hours, all the cells orient in this direction. They're all now pointing this way, okay, in the direction of flow. You change the direction of flow to this. It will take a couple of hours, but they will all orient in this direction. Okay. And the moment you apply flow, signaling, gene expression, everything in the cell changes. Okay. So the mechanoresponsiveness of cells is truly, one of the black boxes that we are just beginning to understand. Right. There is, but it's very, very vital to everything that the cells do in our body. Right. They don't do this without the context of a mechanical cue. We may notice it very obviously sometimes, we may not, but what we are discovering is more and more it is the case that the mechanical milieu, the mechanical kind of cues that the cell is getting is very vital to what the cell, how the cell behaves in your body. Kishan, I took your query, right? Vignesh.
Yes, sir. I was thinking about how nuclei eventually evolved, because when we look at the endosymbiotic theory regarding mitochondria and chloroplasts, we said that bacteria engulf, like archaea engulf bacteria, and then that formed mitochondria. So I read a bit about this, and then there seems to be a sort of similar theory about the nucleus itself. So I was thinking about when initially this sort of endosymbiosis happened and a cell needs to undergo cell division. And during that time, you might not have these centrioles that open up the nuclear membrane and then allow those mitosis phases to occur. So like during those times, would it be plausible to think that maybe the nucleus itself divided usually as a bacteria does, and then
it could be. So it's possible that there existed cells or there existed a bag of lipid in which there were multiple quote unquote nuclei, right? Because essentially it was this original cell that had been taken in and now is essentially dividing. And it may have taken a while for, the particularly, for example, the genetic content of the nucleus driving the control of the rest of the cell is again another huge step in terms of, not just having something inside another cell, but now it having a significant say in what happens. So that's how it could have begun. It's highly possible. We don't know for sure, but the speculation.
You also have the parent DNA and the host DNA, and they like sort of competing each other now. And what happens to the
you're assuming that there was a parent DNA. Doesn't have to be, no. What if this was just the DNA that came in? We don't know, right? We don't know.
Right. Okay.
So it could be just a bag of lipid that took up something, which then became the master of that entire bag, right? And it kind of took over, and now eventually drives everything that that made the modern cell as we know it, right? Yeah, but it's a fascinating area, and there are a lot of questions that remain to be answered. Okay. Anandita.
Yes, sir. I wanted to know if euchromatin and heterochromatin are also part of the chromosomal territories. Is that the same?
Yes. So obviously, there, the U and heterochromatin are where the DNA is wrapped, is present, right? So when you talk about chromosome territories, it's essentially these structures that are kept in place, and they're kept in place. So as I said, you should go look up some of the work that Kundan's lab is doing, because they do very interesting stuff where they're trying to figure out what exactly is in this group of genes or this DNA that is present here that allows it to be anchored to this particular site. Right?
Okay.
Because where it is located, if it's, for example, if and these chromosome territories move around, by the way, so they can be moved around in the cell, it's in the nucleus itself, depending upon what group of genes you want to express how, right? So go look this up. There is a lot of information on how chromosome territories are held, where they are, and how this influences gene expression.
And also, sir, do nuclear pores form and dissociate as and when needed, or is it that they're already formed and they're rigid there?
No, though they form and dissociate. I'm trying to think whether there is a significant amount of the nuclear pore complex that breaks up in a nucleus, regularly. And I'm inclined to say that happens. But let me also look this up. I'm not sure how much of a turnaround happens, but my sense is it does. When cell division obviously happens, there is the turnaround increases pretty dramatically, right? Srijani, your query.
So since physical stress can also, mechanical stress can change gene regulation, and it can probably even cause mutations. So, does the cell have some sort of protective mechanism to prevent that? Because otherwise, all physical injuries or even small changes could cause changes.
Right. So I'm not sure this all cells want to actually not respond to mechanical cues, right? See, mechanical cues will obviously change, expression of genes will affect the behavior of cells. Are cells trying to say, let me not be mechanoresponsive? Maybe there are certain situations where they don't want to be mechanoresponsive, and they may have mechanisms that will prevent mechanoresponsiveness at that time. But cells in general, to large or small extents, like for example, endothelial cells respond to flow in a way that maybe kidney cells don't, right? And the extent of that responsiveness may vary, but it does look like most cells have the ability to react to mechanical cues, right? And they have been doing this all along in ways that we have just not noticed. right? So, for example, tissues have a certain stiffness, and because tissues have certain stiffnesses, the cells, it's a kind of a circular mechanism, or a circular thing, where the cells help create the environment that drives the stiffness, because the extracellular matrix that is secreted by cells, and now that environment comes back and tells the cell how to feel, because of the stiffness that is present in that environment. So there is a feedback loop here, right? And that constitutes part of the normal homeostasis of the cell, right? So, for example, in diseases like cancer, this environment and its stiffness does change significantly, right? And that is thought to contribute to how the cells behave and what they are now capable of doing. So, to kind of answer your query, is there a mechanism by which the cell switches off its mechanoresponsiveness at a certain time? I'm not sure whether it exists in the form that you are asking, right? But it could possibly be happening for specific pathways per se, right? That responsiveness could be changed. We're almost, yeah.
I was actually thinking in terms of how there are some diseases which get triggered after physical injuries. So would they have a
So
genetic
is there a
component?
disease that is triggered after physical injury?
Yes. I know one, there's a chronic pain condition.
No. A chronic pain condition is because of nerves being affected.
Right, but
And
I've
damage.
also heard, so I was reading up on this, and I've also read that they do have a genetic factor to them. So I was wondering if...
No, so see, that's what I'm saying. So those are actually, pain is a completely different mechanism altogether, right? And you cannot associate a physical injury that happens. For example, you fall down, you get hurt, your screen gets scratched, that is injury. Obviously, there is a mechanical damage that has taken place, but that's not driving the behavior of the cell. Everything that happens as a result of the injury, now the cell is reacting to, right? And that's what drives either recovery or not thereof, or lack of recovery, as the case may be.